Hello, and this is my experiment, the effect of telomerase inhibitor IX in conjunction with Bacillus subtilis and or E. coli K12 on pancreatic cancer cells as a cancer therapy. The purpose of this experiment is to create a novel approach to pancreatic cancer research that's both cost-effective, accessible, and has a decreased impact on the body by using microbiological treatments and a chemical therapy. My hypothesis was that telomerase inhibitor IX is applied to pancreatic cancer cells in conjunction with or compared to two types of bacteria, then the cells should have to go undergo reduced proliferation after chemical application, and then undergo apoptosis upon bacterial exposure. Cancer is unregulated cell division with about 4 causes, and it can be either benign or malignant. Pancreatic cancer is very dangerous with a 5-year survival rate because the pancreas is very vulnerable to disorders, especially cancerous tumors, but these tumors don't produce anti-tumor agents, so they're very resistant to immunotherapies. Telomerase inhibitor IX is a chemical that suppresses the tumor proliferation by inhibiting the telomerase enzyme, which basically it stops the cancer cell's ability to divide unchecked. E. coli K12 is a gram-negative bacteria that secretes proteins that are toxic to living cells, and it's widely accessible. Bacillus subtilis is a gram-positive bacteria that secretes fats that inhibit the regulation of a cancer cell cycle. These are my materials. My procedures, basically, I pipetted at the same concentration of cells into 96 wells, and then did varying concentrations of my chemical, my bacteria, or some combination of them, and then measured their absorbance using the Alamar Blue cell viability reagent assay in a spectrophotometer, and I looked at the absorbance so I could see if the cells were healthy or not. This is my table with all of my groups. You can see the chemical, the bacteria, the chemical with the bacteria, etc. My graphs were taken at 24 and 48 hours. These are my absorbance readings after being in the spectrophotometer, and they show significance in all groups under 0.05 after one-way ANOVA. The notable thing here is to see those groups with the stars, as they are the most significant out of all treatment values. It's easier to discuss using this graph because, as you can see, I'd like to take note of the E. coli K12 in the 2 and 4 microliter concentrations, as these had the largest decrease in the proliferation of cells over 48 hours, and that'll be explained why it's important later. Here's some pictures of my experiment. So I did a one-way ANOVA to test the overall significance between groups, and then an LSD test to remove outliers, compare significance, and make the error bars for my figures. In conclusion, I found that my hypothesis was statistically correct, as all p-values were less than 0.05. As I said earlier, you should take note of the 2 and 4 microliter concentrations of E. coli K12 because they cause the largest decrease in viability. The telomerase inhibitor IX wasn't really the ideal treatment because when used with bacteria and used by itself, it still caused smaller decreases in the viability of cells compared to both bacteria. Some limitations were when you have cells in a shaking incubator, it's not the CO2 incubator, so cells are more likely to dry out, which means it created about 4 outliers in my data analysis. I had to do microliter measurements, which are very small, so they have possibility to be skewed, and I tried to take my readings the exact same time every day, but it was a little bit difficult because I'm not perfect. This E. coli K12 is an affordable and equitable cancer therapy, and it's only $15 to $45 vial. It's easily replicable in labs across the world, and the bacteria can't survive in the human body by itself, so it's a very limited effect on the regular human body and the tissues in the surrounding space of the pancreas. In the future, I'd like to have a longer experimental timeline to observe the full effect of telomerase inhibitor IX because it does work over a long period, and I'm afraid that 48 hours may have inhibited the effect of this chemical. And then I'd like to secrete the protein excreted from E. coli K12 to make a more direct cancer therapy so I don't have to introduce a microorganism to a patient. And these are my references. Thank you very much.